This is the lecture for Chapter 18, Practical Applications of Immunology, Part 1. All the practical applications that are discussed in this chapter have to do with antibodies. So the first thing I want to do is review antibodies. The very first thing I talked about was antibody structure. Antibodies are serum proteins, and they're composed of 4 protein chains, 2 identical heavy chains, 2 identical light chains, and every chain has a constant region and a variable region. The constant region is the same for all members of a species, and the variable region of the heavy and the light chain make up the antigen binding site. And the antigen binding site has a particular structure which will bind to the epitope of a particular antigen. So antibodies are highly specific, and every monomer structure has 2 antigen binding sites, so it can bind 2 identical epitopes. B cells are the cells that produce antibodies. So going through the basic life cycle of a B cells are derived from stem cells in the bone marrow. They remain in the bone marrow to mature. When they are maturing, they go through genetic shuffling, which will determine the structure of the antigen binding sites on the antibodies produced by that B cell. Once they have finished maturing, the B cells go to the lymph node where they wait to be activated by an antigen. And those B cells will wait for 3 months, and if they are not activated in 3 months, they will die. But if they're activated, one way to activate the B cells is T independently. That is where the antigen by itself interacts with the IgD molecule on the surface of the B cell. When that B cell is activated, it will go through clonal expansion, divide by mitosis to produce memory cells. Some memory cells, and those memory cells will wait in the lymph node until the second time the person comes in contact with that antigen, and they will wait for 10 years. After 10 years, if they've not been activated, then they will die. The majority of the daughter cells from clonal expansion are going to be plasma cells, and those are cells that start secreting antibody. And the first class of antibody they secrete is IgM, and they will switch over after a couple of days to produce IgG. There are 5 results of antibody binding to antigen, or 5 benefits to the person to have antibodies bind to antigen. First one is agglutination. Agglutination, that involves the clumping of the antibody and the antigen. Then you have opsonization. Opsonization is where the antibody covers the microbe and increases the probability of phagocytosis. Then there is neutralization. When the antibody binds around a virus, a bacterium, or a toxin, that neutralizes those microbes so that they cannot infect the host, or it neutralizes the exotoxin so it doesn't have its negative effect. Also, you have activation of complement, so that is the classical pathway. When complement is activated, you end up with oil, opsonization, inflammation, and lysis. And the last one is antibody-dependent cell-mediated cytotoxicity. This is where the antibody tags a eukaryotic cell so that other white blood cells know to destroy it. The last thing to review about antibodies is the idea of a primary and a secondary response. Primary response is the first time you come in contact with the antigen. So this starts at time 0 when you initially come in contact to that particular antigen. It will take roughly about 10 days to produce any significant antibody in your body, and first will be class IgM that will peak, and later on IgG. And this is your primary response. The important thing about the primary response is that at the end of this, you generate memory B cells. So with memory B cells present, the second time you encounter the antigen, as long as those memory B cells are present, you will have a secondary response, which is going to be much faster and stronger. So in a shorter period of time, you will get significant levels of IgG immediately in addition to your IgM. So the importance of a secondary response is that it is faster and stronger and more likely to clear the infection before you get the disease. So if you mount a secondary response, you are probably immune to that microbe, immune to that disease. The practical applications in this chapter are divided into two types. It's divided into therapies, which are basically treatments for different disorders or diseases, and also diagnostics. So for this part of the lecture, I'm going to focus on the therapies. The first therapy I want to talk about, and possibly the most important therapy, is vaccination. Vaccination relies on this idea of primary and secondary responses. So remember, this is a primary response and this is a secondary response. The idea of vaccination is to produce artificially induced active immunity. So to do that, you inoculate the person with the antigen. So you inject the antigen into the person's body in hopes that their body will produce a primary response. And at the end of the primary response, you generate memory B cells. So if the person actually comes in contact with the antigen naturally, they will immediately produce a secondary response, which will be so strong and so fast, they won't get the actual disease. Jenner was a physician in the late 1700s, and he is credited with the first vaccine for smallpox. And what he did is he would inoculate individuals with cowpox, which is very similar to smallpox. And when he inoculated them with cowpox, they seemed to be resistant to smallpox. And the Latin word for cow is vacca. And that is where the word vaccine comes from. There are several different types of vaccines in terms of what is actually being injected into the person. And what form the vaccine is will have different effects on the level of immunity and the possibility of actually acquiring the disease. The first type of vaccine is a type of whole cell vaccine. And when you say whole cell vaccine, that means you are injecting the whole microbe into the individual. And there are two types of whole cell vaccines. This one is called a live or attenuated vaccine. Live is used for bacterial cells, attenuated for viruses, because remember, viruses are not alive. And for this vaccine, these organisms are alive or active, but they are in a weakened state. So their virulence has been reduced. So they are weakened, but they are still alive. So you are injecting the whole organism into the individual. And this will give the very best immunity because you are injecting all the possible antigens. And remember, every antigen has many epitopes. Also, this gives really good immunity because the microbe is able to replicate at a low level, so it will give the immune system a very good experience at fighting a live microbe. The downside for this, though, is because they are alive, it is possible for a small percentage of individuals to actually get the disease. But usually the disease is a much more mild form. So live or attenuated vaccines will give the individual the very best immunity, but it will cause a certain percentage of individuals to actually acquire the disease. The second type of whole cell vaccine is a killed or inactivated vaccine. So killed, of course, is for the bacterium and inactivated is for the virus. So for this, you are injecting the whole organism, but it has been treated with heat or chemicals to kill it. So these organisms are incapable of producing the disease. They are dead or completely inactivated. So the good thing about this vaccine is there is no way to actually acquire the disease from this vaccine. The downside is that by treating with heat or chemicals, you should know that this will affect the antigens on the outside of the microbes, because remember, antigens are some sort of protein. So by treating with heat or chemicals, that may alter the shape of the antigens. So when you have an immune response to these antigens, that immune response may not be recognizing the actual normal antigens. So the overall immunity is going to be a little bit reduced compared to an attenuated or live vaccine. The next type of vaccine are vaccines from microbe parts. So these are subunit vaccines or acellular vaccines. So subunit vaccines or acellular vaccines, this means you are only injecting the antigen from the microbe, not the whole microbe. So for viruses, it could be protein fibers or spikes that are being injected. For bacteria, it could be any outer protein component. So proteins from the flagella or the fimbriae. It could be glycoproteins from the capsule, the glycocalyx. It could be proteins from the cell wall or other proteins from the cell. So for this type of vaccine, you are only injecting antigens. So there is no way to actually acquire the disease from this vaccine because you are only injecting antigens, but this type of vaccine has the lowest immunity because usually you are only injecting a few antigens, and each antigen only has a few epitopes, so compared to a whole cell or whole virus vaccine, you are injecting many fewer epitopes. A recombinant vaccine is basically a subunit vaccine, but it's just referring to how that vaccine is made. So in this case, they find the gene for the surface antigen. So for this enveloped virus, they could find the gene for the spike, and they put that gene into a plasmid. Then they transform a microbe like a yeast or a bacterium, and then that yeast or bacteria produces the antigen, and they use that antigen in the vaccine. So a recombinant vaccine is basically a subunit vaccine because you're only injecting the antigen. It's just how that vaccine is produced. It's produced using recombinant DNA technology. This is a table that talks about the different types of vaccines for the various diseases. So it lists the disease here in this column, and then it tells you what kind of vaccine. So for measles, mumps, and rubella, it is an attenuated vaccine, attenuated virus, which means it is the whole virus in a weakened form. So it is possible for a patient to actually develop a case of measles, mumps, and or rubella, but it should be a very, very weakened form of the disease. Chickenpox is the same way. So a person who receives a chickenpox vaccine, they could actually develop a very mild case of chickenpox. Polio, rabies, these are killed viruses, so there is no way to develop polio or rabies from the vaccine. Hepatitis is antigenic fragments, so that's a subunit vaccine, so there's no way to develop hepatitis B from the actual vaccine. Influenza. Influenza up here, it has two options. If you have the injected vaccine, it's an inactivated virus. So when you get the flu shot, you cannot get the flu. The nasally administered vaccine, that is actually an attenuated virus. So a person could actually get a mild case of the flu from the nasal spray, but not from the injected virus. A new type of vaccine that they are working on is a DNA vaccine. And I know they are using DNA vaccines in animals, but I'm not sure about people yet. But the idea with the DNA vaccine is that you find the gene for the antigen, you put it into a plasmid, And this time you inject the plasmid directly into the person. So then the human cells will take in the plasmid and they will make the antigen and present the antigen on the outside of the cells. And that way it should stimulate the immune system, and the hypothesis is that you would have lifelong immunity and never need another booster. Herd immunity is probably a term you have heard a lot lately, and the idea of herd immunity is protecting a population from an infectious disease. And this first arose in agriculture, where farmers were worried about protecting their herds of animals, herds of cows, sheep, or goats. They wanted to protect them from the spread of an infectious disease. So that's where this first arose. And of course, humans are basically a herd. We are a group of organisms. So herd immunity is meant to protect those individuals who are susceptible to a disease. So in a group of people, let's say you have a few people who are susceptible to an infectious disease. The idea is if everyone else is immune, then those few susceptible individuals will not get sick. So if everybody else is immune, that means they are not reservoirs of the disease, then they cannot transmit the microbe to the susceptible individuals. Different diseases have different herd immunity thresholds, and the herd immunity threshold is the percentage of individuals in the population that must be immune to prevent an outbreak of that disease. So for instance, diphtheria, you need 85% of the population to be immune to prevent an outbreak of diphtheria. Measles, you need up to 94% to be immune. Whooping cough is another one where you need up to 94% to be immune. So for whooping cough in Winnebago County, in order to prevent an outbreak, we need 94% of the people to be immune. That does not mean that we only need to vaccinate up to 94%. We really have to vaccinate about 100% of the individuals in the area to produce a herd immunity of 94%. And the reason for this is because vaccines have a failure rate. Every vaccine has a failure rate of between 2 to 10%. So if we want 94% of the individuals immune in Winnebago County, we're going to have to vaccinate pretty much 100% of the people. The main reason vaccines fail is something called primary vaccine failure. And primary vaccine failure is a failure to develop a primary response. So in 2 to 10% of the people, this response does not occur. And if you don't have a primary response, then you're not going to generate memory at the end of this response. So when you have the actual exposure to the antigen, it's not a secondary response you're mounting, but a primary response. So the major reason vaccines fail is a failure of the primary response. So a failure of the immune system to respond to the antigen. The reason you get primary vaccine failure is not because there is anything wrong with the vaccine or the antigen. The reason has to do with your own B cells. So remember, during genetic shuffling, it is randomly determined which antigen each B cell is going to respond to. Well, that's random. So at any point in time, the B cells that you have in your lymph nodes, it's random what antigens they respond to. So if you are vaccinated with a particular antigen, it is very possible that at that point in time, you do not have a B cell that recognizes it. Because again, it's just random which type of antigen your B cells respond to. So if you are injected with an antigen and there is no B cell present to recognize it, then you will not have clonal expansion, you will not generate memory cells, and you will not generate plasma cells. So you will not produce any antibodies. And that is why vaccines have a 2 to 10% failure rate in normal, healthy humans. So a normal vaccine will fail 2 to 10% of the time in normal, healthy humans just because it is random which B cells you have present at any point in time. The vaccine schedule for children takes this idea into account. So you'll notice for vaccines such as DTaP, Hib, IPV, PCV13, and RV, a child would get several boosters. And notice that they are several months apart. So when you are giving a vaccine to a child is physically smaller than an adult, has fewer lymph nodes, and therefore will have fewer B cells. So at 2 months, a child is going to have many fewer B cells ready to respond to a particular antigen. So the failure rate for that vaccine is going to be a lot higher in a 2-month-old child. So what they do is a couple months later, they give the vaccine again, because what will happen in those 2 months is that a lot of the B cells that were not activated will start to die off, and you will have new B cells going to the lymph nodes. And those new B cells will respond to different antigens. And then just to make sure your chances are even better, 2 months later, 4 months from the original inoculation, you inoculate the child again, and 4 months later, for sure, you have a completely different set of B cells, and so you are increasing your chances that there is a B cell that will respond to that particular antigen. Because remember, the B cells, if they are not activated in 3 months, they die off. So every 3 months, you get a new set of B cells and more chances to have B cells that will respond to that particular antigen. Another therapeutic application of antibodies is neutralization. So remember, neutralization is one of the results or benefits of antibody binding to antigen. And for this, antitoxins are using antibodies to neutralize toxins. So normally when a person is exposed to a toxin molecule, that toxin, usually an exotoxin, will target a cell, bind to the cell, and cause some sort of damage to the cell. The idea with an antitoxin is that when the person is exposed to the toxin, you immediately inject antitoxin antibodies against the toxin into the person so that those antibodies will bind to the toxin, neutralize the toxin, so there is no effect on the target cell. So that's the idea behind an antitoxin. Some applications of antivenom and antitoxin. First is if you are bit by a rattlesnake, you may get injection of crow fab. And crow refers to the group of poisonous snakes. Crotodilia, and fab refers to the antibody. So in order to generate crow fab, they actually inject rattlesnake venom into sheep, and they are causing a primary response in the sheep, and then they expose the sheep again to the venom to generate a secondary response, and then they purify the antibodies from the plasma of the sheep. So if you are bit by a rattlesnake, you would get an injection of crow fab, which would be sheep antibodies, which would neutralize the neurotoxin before it has its negative effect on your tissues. Another example is if you step on a rusty nail. You step on the rusty nail and then they are worried that you will develop tetanus. And if that happens, they may give you an injection of TIG, tetanus immunoglobulin. So tetanus immunoglobulin, those are antibodies from humans, from plasma, donated plasma, and they would give you an injection to neutralize the tetanus toxin before it interacts at your neuromuscular junction and prevents contraction. Last example is with rabies. If you are interacting with a wild animal that they think might have rabies, then they would give you an injection of human rabies immunoglobulin, HRIG, human rabies immunoglobulin. And again, these are antibodies that will neutralize the rabies virus to prevent the infection. The last therapy I want to talk about is IVIGG, and sometimes this is called gamma globulin. And what this basically is giving by IV an infusion of immunoglobulin gamma, IgG antibodies. And these antibodies are from donated plasma. And this is usually used to treat individuals who are immunocompromised. Possibly they are undergoing chemotherapy or they have other conditions where their immune system is depressed. And so giving them an infusion of IVIGG, this is a form of artificial passive immunity. So it's very good at protecting them from a lot of infections, but because it's passive immunity, there is no memory, so it's not long-term.